Now that we know what an action potential is and how the cell generates this action potential, let's discuss how the action potential actually propagates or moves along the axon of our nerve cell. So let's begin by recalling some basic information about the neuron. So basically, the neuron contains the soma, also known as the cell body, that contains the nucleus and the organelles. It contains these projections known as dendrites that receive the electrical signal from other cells, and then our dendrites sends those electrical signals through the soma and to the axon hillock. Now that axon hillock receives the stimulus, and if the stimulus is high enough, if it reaches the threshold value, an action potential is generated on the axon hillock, and then that action potential somehow moves along the axon and all the way to the axon terminal away from the soma of the neuron. Now, the question is, how exactly does our action potential actually travel along the axon, and why does our action potential only move in this direction away from our soma? So let's begin by zooming in onto our axon hillock as shown in the following diagram. So this is before any stimulus is actually applied. So before we apply our stimulus, the inner portion of the cell membrane, so this is one cell membrane on this side, and this is the other cell membrane on the other side, and this is our cytoplasm of the axon hillock. So before we apply our stimulus, our cell membrane is resting. And remember, the resting membrane potential means that the inner portion is negatively charged and the outer portion, the extracellular portion of the cell membrane, is positively charged as shown. So before any stimulus is applied, the membrane is negatively charged inside the cell and positively charged right outside the cell. Now, let's suppose our dendrites receive that signal and transmit that signal through the cytosol of the soma and to the axon hillock. Now, if the stimulus is high enough, if it is equal to or exceeds the threshold value, then what that basically causes is the opening of the sodium voltage gated channels. And as soon as our stimulus is applied to a certain region, let's say this region, our channels, the sodium channels on the membrane open up, and the sodium travels down its electrochemical gradient and from the outside and into that cell. So we have an influx of sodium into our cell. Now, as soon as our sodium ions rush inside the cell, they will reverse the polarity of the cell, and this is known as the depolarization period. So that basically means because we have positively charged sodium ions flowing into the cell, that will make the inside of the cell momentarily positive, and the outside of the cell will become momentarily negative in the region where the stimulus is applied. So that is shown in the following diagram. The sodium channels open up and our sodium ions move into the cell as shown, and that will cause the inside of the cell membrane to become positively charged and the outside to become negatively charged. Now, notice the adjacent region of the cell membrane still contains a negative charge on the inside and the positive charge on the outside. The question is, how exactly will the adjacent section of the cell membrane be influenced by the influx of the sodium ions into the cell? So it turns out that the increase of the positive charge inside the cell where the stimulus actually took place will cause an increase in the positive charge found in the region right next to where the stimulus actually took place. So what that means is, as we have a buildup of sodium ions in this region, that will influence the charge value on the region right next to it. And so this will begin to become more positively charged. And remember, as the inside of the cell initially becomes more positively charged, that will begin to stimulate the opening of the sodium ion channels, the sodium voltage gated channels. So what that basically means is we stimulate the Na channels to open up adjacent to where the stimulus actually took place. So to see what we mean, let's take a look at the following diagram. So as soon as the sodium channels begin to open up on the adjacent region, the sodium channels where the stimulus actually took place begin to close. And as they begin to close, that will basically inactivate our sodium channels, and that will open up the potassium channels. And now potassium ions, where the stimulus actually took place, the potassium channels will begin to open up, and our potassium ions will begin to move down the electrochemical gradient from the inside of the cell to the outside of the cell. And this is known as our repolarization period. Now, we're going to once again change the polarity of the cell, and so the inside will become negatively charged because we have these positively charged potassium ions leaving. So that means the inside will become negatively charged, the outside will become positively charged. But at the same exact time, the concentration of positive charge increases in the adjacent region, and that causes our sodium voltage gated channels to open up in the adjacent region. And so we have an influx of sodium ions into the cell around this region. And in this manner, we can see that the action potential moves from this initial position where the stimulus took place to the next position along the cell membrane of our axon. So once again, at around the same time as the sodium channels begin to close and the potassium channels begin to open in the stimulus region, in this region here, the sodium channels in the adjacent region will begin to open, causing depolarization to take place here. So we have depolarization taking place here and repolarization taking place here. Now, in this same effect, so we basically have a domino effect taking place. And so each adjacent consecutive region will basically be depolarized as our action potential moves along our cell membrane. Now, the next question that we want to basically answer is, why doesn't our action potential move in the opposite direction? So we said earlier that as the positive charge increases on this side, this inside negative charge becomes more positive. And that's exactly what causes this section to depolarize. And the same thing is true here. So as this inside becomes more positive, this inside also begins to become more positive. And so as these ions begin to close, these sodium ions will begin to open. And so the action potential will begin to move in this direction. But notice in this case, we also have a negative charge to the left of this increasing positive charge. So in the same way that this negative charge becomes more positive when we have the influx of sodium, why doesn't this, the one right and back, also become more positive? And so we also have an action potential traveling this way. Why doesn't that actually take place? Well, it turns out that this region that is being repolarized is actually experiencing an absolute refractory period. And that means that all the sodium gated channels are actually closed. All the sodium voltage gated channels are actually closed and inactivated. And that means no matter how high the stimulus is, no action potential can be generated in the region right before our action potential. So once again, you might be wondering why the action potential does not move back towards our cell body. So we know it moves this way, but why doesn't it move backwards? Well, this is because the region of the membrane right before the depolarized section is in its absolute refractory stage. And that means no matter how large our stimulus actually is, no action potential will take place in that that region, and this is because the sodium channels are inactivated. There's nothing we can do that will activate those inactivated channels. So we have to wait until they recover from that inactivation period. So this is explained in the following diagram. So as our action potential moves over this way, the region right in back of that action potential is experiencing a refractory period, absolute refractory, and that means our sodium channels on the cell membrane here and here are inactivated, and no matter how high our stimulus is, no action potential can be generated in this section, and so our action potential does not actually move in the backward direction towards our soma. It only moves towards our axon terminal, and this is shown in the following diagram. So our stimulus is applied to this section. If it's high enough, we basically have this propagation of the action potential take place, and it only moves away from the soma, away from the cell body, and towards our axon terminal. So this is the mechanism by which our propagation of the action potential actually takes place. In the next lecture, we're going to discuss a concept known as myelination. We're going to discuss what makes our signal propagation faster on the axon, and we're going to discuss something called saltatory conduction.